right?
Yeah.
Okay, can you see the screen?
Yes, ma'am.
Yes.
All right. Okay, students, switch on your videos. Let's start today's class. So in the previous class, we had discussed about color blindness, right? How color blindness inheritance takes place. We know that it is like an X-linked recessive disorder, meaning that it will express itself in a very homozygous condition and it will not express itself in any other condition. Now, if, for example, XC, this is the gene that is being carried by the, or it is the faulty gene, right? It is the gene that carries the color blindness. Now, those organisms that only get one, not organisms, the humans that have this genetic predisposition of having XC along with a Y. So that would be a male because XY is a male, and this male would be affected. Now, since it is an X-linked recessive disorder, so here that one X is going to be the determining factor in the case of males, because this X is only going to tell us whether the male is affected or not, because this disease is not Y linked, it is X linked. Then comes XC condition wherein the female is affected. And then the third condition is XC and X, that is when the female is a carrier of this particular disease called as the color blindness. Meaning, if any woman or if any person is a carrier of a particular disease, then that means that she will not have the disease, but will have the potential to transfer the disease to the future generation. So this follows a crisscross inheritance pattern. During this, what happens? A male transmits his trait to his grandson through the daughter, and a female would transmit it to the granddaughter through her son. It's not a direct transmission like from a parent to the son or to the daughter. It is a crisscross inheritance. Now, in this disease, the person who's affected with this particular disease does not distinguish between the red and the green color. Okay, they have trouble seeing the color. Okay, so now let us see what are the possibilities and how the inheritance of color blindness can take place. The first condition is when a woman is normal, whereas the male is a colorblind male. So in that case, these are the gametes wherein you would find that there are two carriers, two daughters that are the carriers of this particular disease, and then two sons that are normal. In this case, wherein a normal woman marries a colorblind man. The second case is when a carrier woman, meaning a woman that has the gene for color blindness, is marrying a colorblind man. So in that we saw that in this case there will be one affected female, two affected males, and one carrier females. Then we talked about hemophilia. Did I discuss hemophilia in the previous class? Yes or no? Girls, are you there?
Yes, ma'am.
Switch on your videos, students. Quick, quick. Now talking about hemophilia. So hemophilia is an X-linked recessive trait. Meaning again, the inheritance of this particular disease is also just like how it is for color blindness. Now during the disease or in the disease of hemophilia, what happens? The blood clotting factor is defective. So the person who has this disease will not be able to clot his or her blood during the case of emergency or during the case of injury. That won't be happening. So now talking about the way it has been transmitted. So XH is the genotype of the hemophilia, and you can see that XHY. This case wherein the heterogamety condition is there, in this the male is affected. Whereas in the second case, the female becomes a carrier or the female becomes affected. These are the possibilities. Now, the possibility of a female becoming hemophilic is very rare because the mother of such a female has to be at least a carrier. The mother has to be a carrier. Along with that, the father should be hemophilic. Then only like you can say the female can become hemophilic. Now, why is this rare? Because a male that is hemophilic will not be able to survive, or he is unviable. So it's of no use, and it's a very rare case wherein the female would become hemophilic. Also, the females that are actually hemophilic, they will die before the birth. So in this case, when a normal man marries a carrier for hemophilia, now you people can make out the crosses, how that can be, like how the transmission of that particular disease can be done. Then comes the third disease, that is sickle cell anemia. Now, during this case, see, there are two genes, HbA and HbA. Now, this is also a homozygous condition. So, HbA, it is a normal condition. It is homozygous, but it is a normal, meaning the person who has this gene is not affected. But later on, if HbS is the gene that is for sickle cell anemia. So, if HbA and HbS is there, so the human is normal, but it is a carrier of the disease. Then comes HbS and HbS. So here the person is diseased and they die before attaining maturity. Now this HbS is a mutated gene, meaning mutation took place over here. Now if you might remember, we talked about substitution mutation, wherein a purine was being substituted by a purine called as transition. Pyrimidine was being substituted by a pyrimidine called as transition. But when a purine was being substituted by a pyrimidine, we called it as what? Transversion, right? We have studied this mutation. Now, in this case, what happens? In this case, transversion happens, meaning a purine gets replaced by a pyrimidine. So this disease is caused by mutation, that is transversion of the gene controlling beta chain of hemoglobin. So the mutated gene is now HbS. Normal gene is HbA. So HbS causes one change in the amino acid sequence of beta chain. What happens in the case in the chain beta chain of globin? The glutamic acid is replaced by valine at the sixth position of the beta globin chain. This is very important. Which position and which amino acid is being replaced by another amino acid? So if we talk about glutamic acid, that is being replaced by the valine. Now the mutant, now since it's like a wrong sequence, wrong amino acid is present. So the mutant hemoglobin, it undergoes polymerization. It starts forming a polymer under low oxygen tension, and then the shape of the RBC from biconcave disc-like shape, it changes to elongated sickle-like structure. Okay, it becomes elongated from that. And now this means oxygen carrying capacity decreases because the size becomes smaller. So how will it carry enough oxygen? Then comes phenylketonuria. Now, phenylketonuria is yet another disease, and in this case, metabolism is faulty. Now, remember that this is not an X-linked disease, rather, it is an autosomal recessive disorder, that it will be expressed in a homozygous condition, but it is autosomal, meaning autosomes are involved, meaning the body cells are involved. The sex cells have nothing to do with it. Now, the individual that is affected by phenylketonuria, they lack the enzyme called as phenylalanine hydroxylase. This phenylalanine hydroxylase enzyme is very important for conversion of the amino acid that is phenylalanine into tyrosine. But if this enzyme will not be there, then phenylalanine will not turn into tyrosine, because of which it starts getting accumulated. So the phenylalanine accumulation takes place, and then after the accumulation is done, it leads to the conversion of phenylalanine into phenyl pyruvic acid. And when this phenyl pyruvic acid accumulates in our body, it causes mental retardation. So that is why this particular disease called as phenylketonuria is very, very harmful. It's sort of lethal. And it can also, it has such an adverse impact on the human mental retardation. Then comes thalassemia. Now, thalassemia is yet another autosomal recessive disorder. Now, this is also happening because of mutation, particularly deletion mutation, meaning one gene gets deleted. Now, in this case of thalassemia, what actually happens in this disease is that the synthesis of the globin chain of RBC, we know that hemoglobin is present. So, hemoglobin is made up of heme and globin. Heme means the iron part, and globin is the protein part. So, here the globin, meaning the protein part, gets defected and abnormal hemoglobin is formed. Now, this thalassemia disease, wherein abnormal hemoglobin is formed, it is of two types, alpha thalassemia and beta thalassemia. Now, remember that the hemoglobin chain has two types of, you can say chains or cells, alpha and beta. So, accordingly, two types of thalassemia are there. When we talk about the alpha thalassemia, so here the production of alpha globin chain is affected. In the case of beta thalassemia, the production of beta globin chain will get affected. Now the genes, if we talk in terms of genes, so HbA1, HbA2 genes present on the chromosome number 16, meaning the genes for the preparation of proper hemoglobin is present on the chromosome number 16. So that gets involved. And then HBB gene that is located on the chromosome number 11 controls the beta thalassemia. Any doubts?
No, ma'am.
Now moving forward, there is sort of like a comparative analysis in between the differences of thalassemia and sickle cell anemia. So in this case, because both of them are related with the globin chain of hemoglobin, but how are they different? In the case of thalassemia, it is a quantitative problem where too few globin molecules are synthesized, meaning the globin molecules are synthesized, they are not enough, meaning they are very little, very less amount. And sickle cell anemia is a qualitative problem. Qualitative means the quality of the globin chain is affected here. It is not related with the number, rather it is affected with the property, that is the quality. So here incorrectly functioning globin chain is synthesized. So this is a major difference in between thalassemia and sickle cell anemia. Now talking about genomatic mutations. So we did talk about the normal mutations. Now let us have a quick overview as to what are genomatic mutations. See, whenever the number of chromosomes change, there is like a change in the whole organism's phenotype. Why? Because every chromosome has a certain function to perform, and every single chromosome is responsible for every single phenotype of our body. So in this case, the genomatic mutation is the change in the chromosome number that brings about visible effects on the phenotype, meaning the external appearance. Now, the genomatic mutation can be of two types. There is aneuploidy and then there is euploidy. Now, aneuploidy has two things, that is monosomy and trisomy. Euploidy has haploidy, polyploidy, diploidy, meaning haploidy is only one set of chromosome. Diploidy is two sets of chromosomes. And when more than two sets of chromosomes are present, we call it as polyploidy. Now we know that the normal chromosome in us humans for a diploid cell is 46, that is 23 pairs. And we know that whenever we write about the 23 pairs, we say that 22 of them are autosome plus X or Y. If it is X, then 22 plus X becomes 23. If it is Y, then 22 plus Y becomes 23. So this is how we describe. We know that this is autosome, meaning the body chromosome, and these are the sex chromosome. That is how we differentiate them. Now, in the case of monosomy, 2n minus 1 takes place. Monosomy means one of the chromosome is less. Nullisomy means two chromosomes less. And then trisomy is increase in the one number. For example, if normal human has 46, so after trisomy, it becomes 47. So that is how it happens. Any doubts up until now, students?
No, ma'am.
Okay. Now coming on to the last topic of this chapter, that is chromosomal disorders. Chromosomal disorders. Now, they are very different from the Mendelian disorders. When we talked about Mendelian disorders, we saw how genes, like because of the effects of genes, what are the particular, you can say, diseases that are caused in human beings. But right now, we're going to talk about these chromosomal disorders because of the variations, or you can say changes or imbalance in the chromosome number. The first is Down syndrome. All of you have heard about this. This particular disease was described by Langdon Down in the year 1866. Now, this disease occurs due to trisomy of chromosome number 21. Trisomy means one extra chromosome is present. 2n means 46 chromosome diploid condition. Plus 1 means like one extra chromosome is there. So, total 47 chromosome this person has. Now, what happens in this particular disease? Short stature. The person who has this disease is usually of short height. Small round head, furrowed tongue. The spelling of tongue is wrong here. So, furrowed tongue is present. The palm is broad and a lot of creases are there in the palm. Crease means lines. Then big wrinkled tongue is also there. Physical, psychomotor and mental development is retarded. Physical means the genitals, the gonads, all of these will not be developed properly. They'll be poorly developed. So this is about the Down syndrome. One more, you can write that they have almond shaped eyes. Almond shaped slant eyes. Slanted eyes. Then comes the next disease, that is Klinefelter syndrome. This one takes
Ma'am,
place in the case of, yes,
what does furrowed tongue mean?
furrowed tongue means like just like how if you open your mouth, your tongue stays inside. But for them, the
Yeah.
tongue is always protruding out. They can't
Oh,
control their tongue to keep it in their mouth. That is furrowed. Okay.
okay.
Now talking about the Klinefelter syndrome. So in this case, this Klinefelter syndrome takes place in the case of males. Right? It is like not there in the case of females. It is not affecting females. Now in this case, what happens is that there is the presence of additional, you can say, X chromosome in the males. Normally, males have one X chromosome and one Y chromosome. But in this case, meaning in the case of Klinefelter syndrome, the male has an additional X chromosome. So, what happens and how does this happen? So, for example, if during the time of fertilization, abnormal egg, meaning which has an extra X chromosome, was fusing with a normal sperm, then also this disease can be caused. Or when a normal egg fuses with an abnormal sperm, then also this disease can be caused. Now, like the people, the males that are like they have the Klinefelter syndrome, or they are all sterile, meaning they can't reproduce. Okay, they can't reproduce. And uh, students, what is the opposite of sterile? Tell me.
I don't know the exact word, but sterile means they can't reproduce.
Yeah, you are right. So, what will be the
There's
opposite
a
of sterile?
reproductive.
You people know the term. Just try
Oh,
to recall it and tell me. You know it. You have studied it. You have heard this term a lot.
does
So, tell me.
is
Sterile
it
means a
similar to this word?
sort of sounds the
Okay.
same. Like
Okay.
sterile, the sound is same. The words are
Okay.
different. Try to recall.
Mm,
Sterile means they can't reproduce. And the people that can reproduce are known as?
Fertile?
Yes,
Oh, like,
fertile.
oh,
Very good. Very good. Who said the answer?
me, ma'am. Like,
Okay, very good. All right. So these people that have the Klinefelter syndrome, they are sterile males. They can't reproduce. They have a masculine development because they do have the X chromosome, but because of the presence of additional X chromosome, they have certain female characteristics in them, such as feminine pitched voice is there, development of breast that is known as gynecomastia, not functional breasts, but the size of their breast, the chest increases because of the presence of an extra X chromosome. This much is clear to everyone.
yes,
Now
ma'am.
coming on to next and the last, I think this is the last disease. Yeah, there is. Oh, pedigree analysis. Okay. The last is the Turner syndrome. Now, Turner syndrome, it is the disease that is caused in the case of females. And here monosomy takes place, meaning 2n minus 1. Normally, a female is supposed to have one X chromosome, right? And one X chromosome from thing, uh, the father and one from the mother, right? That is how a female is made. But in this case, when an abnormal egg, meaning the egg that has the body chromosomes but that does not have the X chromosome. When that type of egg fuses with a normal sperm, or when a normal egg fuses with an abnormal sperm. So, in that case, a Turner syndrome condition takes place, and in this case, the female is sterile. Why? Because their ovaries do not develop, because they do not have sufficient X chromosomes in there. They have very underdeveloped body structures. They have rudimentary. Rudimentary means not functional, poorly developed. And they also do not have secondary sexual characters, meaning like feminine, like all of the things like menstrual cycle, these things do not happen in those fields. This much is clear. So chromosomal disorders are clear to you all, right? I hope.
Yes, ma'am.
Yes, ma'am.
Okay, good. The last thing is pedigree analysis. Actually, this is there like in the middle of the chapter only, but I think I did not mention it in the middle. So at the last, see, pedigree analysis. This is like, have you people drawn family trees when you were younger? Paste pictures like grandparents, parents, children. Have you done family
Yes,
tree
ma'am.
project? All of you have, right? I think all of you have. So pedigree analysis is just like a family tree only, but it is darker version of family tree. Why? Because this is going to tell us the genetic disorders, meaning how the inheritance of a particular disorder took place. Like with the help of pedigree analysis, people can find out whether like for example, any human being has a disease. So whether this disease is coming from the parent, whether it is coming from the grandparent directly, that can be analyzed. So it is a method of study of human genetic disorders. The analysis of traits is done in several generations of a family, and we call it as pedigree analysis. Now, inheritance of a particular trait is represented in a family tree over generations, and they can be helpful in detecting the disorders such as the Mendelian disorders, like the disorders of all of these cases, hemophilia, color blindness, all of these disorders can be detected by pedigree analysis. Now, there are some symbols that are used to do the analysis. The square is to represent the unaffected male. The circle, like this square, normal square with nothing in it, is used to represent the unaffected male. The circle with nothing in it is used to denote the unaffected female, and the diamond shape is when the sex is not known, whether it is a female, whether it is a male, but this is also unaffected. Then whenever the squares and circles and the diamonds are filled with, like they are shaded or they're colored, then that is denoting affected individual, affected male, female, and whoever is the sex unspecified, that is also affected. When a line is drawn between a square and a circle, meaning a male and the female, both are unaffected, so that is known as mating. When two lines are drawn, then that shows mating between relatives. And This is how the pedigree analysis is done. Like this is the father, this is the mother, and this is the daughter and the son. Pedigree, like parents are present above and children are present below. And normally it is read from left to right, meaning if any symbol is drawn at the left, it means that that particular individual has been born first. Then comes the next. This second depiction shows that parents with male child affected with a disease, right? And then the last one is like whenever this diamond has 5 written in it, it means that 5 unaffected offsprings are there. When it is colored, then that will mean 5 affected individuals or offsprings are there. Now, by pedigree analysis, one can easily understand whether the trait is autosomal dominant, whether it is autosomal recessive, sex linked. All of these things we can find out with the help of pedigree analysis. Now, in the case of recessive allele, characteristic condition can appear in the progeny of unaffected parents as well. Two affected individuals cannot have an unaffected child. It's inevitable. If the parents are affected, then the child will be affected. And if the parent is a carrier, then there be a possibility that the child will not be affected. But if the parents are affected, then the child has to be affected. It can't be unaffected. Now, the different types of inherited diseases: autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive. All of these you have already studied. So, with this, we have completed the chapter. Now, tell me if you have any doubts up until now. Hmm? No doubts? Okay, then
No.
tell me what is the last thing you people have written so that I'll give you exactly that to take down the notes. What is the last thing you people have written? I think you have written hemophilia, isn't it? Check and tell me quickly. You people are so slow to reply. Reply quickly. Check in your notebook and tell me. Most of you have not attended the class only. So Amna, you only tell me.
Ma'am, colorblind.
Color blindness. Okay, color blindness was completed, right?
Yes, ma'am.
Okay, now you people have to note down from hemophilia. Okay, now you people can note it down. Tell me once you're done, so I'll scroll. And try to write a little faster because you have less time.
Done, ma'am.